Thank you so much, Brandon, and for organizers, Andrea, Stephen, and Mark, for inviting me. It's a great pleasure to be here and present in this conference, or in this little special session. I could have given the title of my talk as my research under the guidance of Michael Levitt, because I basically collected all the work we did together with Michael. Actually, even the word, the phrase called natural modeling came from Michael too. Interestingly, I want to start with some of the challenges in experimental structural biology, and also some trends that we have been seeing that the larger and larger structures coming out. Unfortunately, resolution is lower and lower, and that's the price we are paying for it. However, this gives tremendous potential for us computational structural biologists trying to aid, facilitate, and interpret experimental data. But we do have to face our own challenges because since there are large systems, we have multiple scales. If you want to model there is an example here from antibody. You could see that there are CDR regions, Fab regions, and the entire Y shape of the antibody. There are different structural motions occurring at different levels, and it's a big challenge to model them. And when you try to do that, you are going to experience some problems, and some of them are related to the large number of degrees of freedom we have to deal with. And here's a little case study to illustrate this problem. Supposing that you want to evaluate the relative orientation of two helices here, and it's quite evident that there are 6 degrees of freedom is enough to describe this kind of structural space. However, if we start with the helices, assuming that all have like 100 residues roughly, and using a bottom to top approach, using all the atoms and maybe something called molecular dynamics, we end up spending or using three orders of magnitude more degrees of freedom, roughly like 6,000, and hoping that this kind of essential motions which describe the relative orientation of these two structural units are going to emerge. Unfortunately, it doesn't. And that is the reason we invented a technique which I would call here natural move Monte Carlo. And the rest of my talk is going to be about this little tool we developed together with Michael. And this was one of the first method paper we wrote together. In natural move Monte Carlo, we can explicitly incorporate like collective moves into the system. So here is a little demonstration here with a helix and a 3-stranded beta sheet. We can move the two structural units with respect to each other. Unfortunately, when you do that, you are going to break the interconnected chain between these units, but we developed a very efficient so-called linear complexity chain closure algorithm to handle this problem. Similarly, for nucleic acids, one of the most natural choice for degrees of freedom moving or different base pairs independently, and all the corresponding chain breaks are going to be processed similarly with our new chain closure algorithm technique. And now I just want to focus a couple of applications we did together with Michael in the past years. One of them was, as I mentioned at the very beginning of my talk, to interpret experimental data, cryo-EM spectroscopy data on, in particular, a very well-studied system, chaperonin, MMC bacterial chaperonins, and this particular molecule or molecular machine has like 2 chambers, and they are basically folding proteins. The 2 chambers have 8 subunits each, and each subunit itself have like 3 domains: an apical, an interior, and an equatorial domain. And this is a system where you could it's a very good benchmark system to test the multi-scale nature of these natural degrees of freedoms, because you could start with the highest coarse-grained degrees of freedom, moving each unit independently, then the domains within the units, then different structural segments within domains, and so on. So this approach is actually a top to bottom approach. We are dealing with the, focusing on the most collective structural motion, and in order to facilitate that, we are introducing finer level degrees of freedom. The similar technology we applied for RNA junctions, which appear in many molecular machines. First, I'm just showing you here a very simple, interesting benchmark example we did to try to assess the accuracy of the method in order to sample the empire conformational space. We basically created a fractal-like RNA by fusing together four four-way junctions, meeting in a central four-way junction. And in this system, we are lucky enough to know that, for example, if you look at the n distances of adjacent four-way junctions, they are going to be identical. So the four distribution should coincide. It is a necessary requirement that they all converge to the same limiting distribution if you sample the entire space correctly. And as a proof of concept, as we have been introducing higher and higher degrees of freedom with higher level complexity, the four distribution starts to coincide. This was a very efficient way of testing this algorithm. And we applied this, this kind of method to test even experimental observations. People have been interested these days in building interesting structures out of naturally occurring tRNAs. I would call it a tRNA LEGO. And one is interesting structure is an RNA cube, which could be built out of two RNA squares, which you could create if you fuse two naturally occurring tRNAs together. And interestingly, in a wet lab experiment, they noticed that there are certain mutations in the tRNAs affecting the yield towards the tRNA square formation. And with our technology, we managed to test the effect of these mutations, and what we found is that the corresponding mutations are significantly changing the angle distribution between the helices. So in the mutants, while in the wild type, the angle distribution is very close to the right angle, which gives the high yield towards the RNA square formation. Once you introduce two mutations, you see that the angle distribution are significantly off from the right angle. And that is an explanation why your yield towards RNA square formation is going to lower. And now the last application, which we recently did with Michael, we used natural move Monte Carlo in a protocol to predict primary chromatin structure. And the protocol itself takes a query sequence which could be tens of kilobases long, and we are sliding through a sequence window on this query sequence, one nucleotide at a time. The sequence window indicates a local sequence which we could thread on a high-resolution nucleosome template, creating a structure. In that way, each local position of this nucleosomal position can be associated with the nucleosomal structure. We could use natural move Monte Carlo, evaluate a particular energy of the structure. In fact, we used the energy difference between the sequence occupying the nucleosomal form and minus the energy of the same sequence, assuming a linear form, and we call this energy the nucleosome formation energy. And what we managed to show that if we use this protocol, and then we can actually get some interesting correlation with experimental results predicting nucleosome occupancy. You may have heard about nuclease digestion, MNase digestion experiments. They can produce you profiles, and here is a profile on a well-studied region from yeast chromosome 14. the red line indicates a nucleosome occupancy profile, and the blue line is our nucleosome formation energy for different sequence positions of the nucleosome. And as you could see, wherever the occupancy is high, our nucleosome formation energy is low. The overall correlation is roughly 0.67, meaning that this is basically what we predict as being the physical factor in nucleosome positioning. And finally, as a futuristic application of this technique, we hope that we could move beyond the primary chromatin structure and even predict, using like atomistic and training-free methods, so we could predict higher order chromatin structure as well. And as a last point, our atomistic nature of the technique allows us to also capture epigenetic effects, either natural or the ones which has been used by epigenetic drugs. And finally, I would like to mostly thank Michael for all his generous mentorship and some other postdocs and students involved in this project. Thank you so much. Do we have time for questions, or we don't? Okay.
Hi, I was just wondering about your tRNA squares, whether
Yes.
those mutations that you mentioned, do you find that in nature they're selected against? Does that make any sense to you?
Actually, we basically reproduced an experimental observation. We haven't really looked into the nature of these mutations. This was a kind of proof of concept whether we could correlate our completely atomistic and virtual technique to something in a wet lab.
Then that might be interesting
Yeah,
to
it
look
might
for,
be an
because
interesting thing to look at.
if that were like an artifactual, an artifact that needs to be avoided, or...
Yeah, definitely. So the only point what I wanted to make is that with these kind of techniques, we have some kind of potential to predict what may happen, right? You don't have to do everything in a wet lab. Make a mutation, gene distribution, so we could scan many different possibilities and aid and facilitate nanostructure design in the RNA world.
So I was just curious, you talked about how your technique allows these natural moves. Yeah. And I was just curious, does it also allow a move that is, in a sense, very unnatural or very strange, but is very efficient or very useful for Monte Carlo? Like you wouldn't think like a thing would normally move like this, but it turns out very useful for searching in Monte Carlo.
So basically, that's a very good question. I showed you a lot of systems where the choice for the move could naturally emerge from system symmetry considerations. However, one natural choice which is hard to object is that if you take all the secondary decomposition of a structure into segments, or even supersecondary, like entire beta sheets, that you can take these structural segments and first assume that they are rigid and assign a three-body rotation and translation to each. So our technique is basically a tool set for a hypothesis-based modeling where somebody with intuition or experimental knowledge can come in and say that this is a hypothesis, I believe that these degrees of freedom are important. Then we could give you a software which could evaluate all distributions along the degrees of freedom you would like to use. So rather than saying that these are the right degrees of freedom, I would say that what we have is a hypothesis-based modeling which needs an input from experimentalists. Apparently, it turns, I'm a method developer. Experimentalists sometimes are working on a structure for 20, 30 years. They know everything about the structure. So what we hope that our tool can bridge the gap between the experimental work and all the knowledge accumulated there could be now used and efficiently explored.